Hello students, we have learned about linkage, recombination, sex determination and mutation. I hope you have understood about them. Now we are going to have Mendelian disorders, chromosomal disorders and pedigree analysis. Now let us learn about genetic disorders. What is a genetic disorder? Genetic disorder is illness or disorder which is caused by DNA mutation. You know, genetic disorder is of two types: Mendelian genetic disorder and chromosomal genetic disorder. Mendelian disorders. Mendelian disorders are caused due to mutations in single genes. They are transmitted from parents to the offsprings as per Mendelian principles. How it is different from chromosomal disorder, let us see. Chromosomal disorders are caused due to absence or excess of abnormal arrangement of one or more chromosomes. Chromosomal disorders are not inheritable. You know, Mendelian disorder is again of two types: autosomal disorders and sex-linked disorders. Autosomal disorder is again of two types: dominant autosomal disorder and recessive autosomal disorder. Sex-linked disorders may be of Y-linked and X-linked. X-linked disorder is again of two types: recessive X-linked disorder and dominant X-linked disorder. Some examples of dominant autosomal Mendelian disorders are myotonic dystrophy, achondroplasia, polydactyly, Huntington's disease. Some examples of recessive autosomal Mendelian disorders are albinism, sickle cell anemia, thalassemia, cystic fibrosis, phenylketonuria. Now X-linked. Let us know some examples of recessive X-linked Mendelian disorders: hemophilia, red-green color blindness, protonopia, tritanopia, deuteranopia, muscular dystrophy. Now let us study about some X-linked disorders. Firstly, hemophilia. What is hemophilia? It is an X-linked recessive disorder. This defect may come from a carrier mother and a normal father to their son. This disorder is also known as bleeder's disease, as the patient will continue to bleed even from a minor cut, since he or she does not possess the natural phenomenon of blood clotting. This disorder, hemophilia, is due to presence of a mutant gene X-ICE, that is a part of the cascade of proteins antihemophilic globulin or plasma thromboplastin involved in the clotting of blood. It is common in males because male has only one X chromosome and the Y chromosome does not have the conjugative gene. It is very rare in female because mother of such a female has to be at least carrier and father should be hemophilic. It is fatal in females. Queen Victoria was a carrier of this disease and passed on the disease to some of her sons. Next, let us learn about color blindness, which is also a recessive sex-linked disorder. You know, it is a recessive X-linked trait in which the eyes of the individual fail to distinguish the red and green colors. The recessive allele are carried by X chromosomes. In female, it appears only when both the X chromosomes carry the gene XC. The females function as carriers in presence of a single recessive gene XXC. In males, the defect appears in presence of a single recessive gene XCY, because Y chromosome does not carry any gene for color vision. Okay, now let's have some recessive autosomal disorders or Mendelian disorders. Firstly, sickle cell anemia. It is caused by a mutant recessive allele on chromosome number 11. The mutant gene causes the substitution of glutamic acid by valine at the 6th position of the beta globin chain of hemoglobin. You know hemoglobin has 4 chains, 2 beta chains and 2 alpha chains. Beta chains of hemoglobin are having 7 amino acids. In the 6th position of the beta chain delta, glutamic acid is present. If the glutamic acid is changed by valine by mutation, point mutation, the beta chain of the hemoglobin become defected and the individual will be affected by sickle cell anemia. The defective hemoglobin undergoes polymerization under low oxygen tension and changes the shape of RBC from biconcave cells to sickle shape or elongated cells. The disease is controlled by a single pair of alleles, ISBA and ISBS. Individuals homozygous for ISBS show the disease. Though heterozygous individuals ISBA, ISBS are carriers because it is a recessive disorder. Okay, now let us have some idea of thalassemia, which is also an autosomal recessive blood disease which appears in children of two unaffected carriers. The defect can occur due to mutation or deletion of the genes controlling the formation of globin chains, commonly alpha and beta of hemoglobin. Imbalance synthesis of globin chains of hemoglobin causes anemia. Depending upon the globin chain affected, thalassemia is of 3 types: alpha, beta and delta thalassemia. Alpha thalassemia is caused by the defective formation of alpha globin. The latter is controlled by 2 genes present on chromosome 16, ISBA1 and ISBA2 with a total of 4 alleles. There is anemia, jaundice, hepatosplenomegaly and bone changes. Beta thalassemia is caused due to alleles of ISBB gene present on chromosome number 11. Delta thalassemia occurs due to defective alleles of ISBD gene present on chromosome 11 that forms delta chain of hemoglobin. Thalassemia is quantitative problem and sickle cell anemia is a qualitative problem of defective functioning of globin molecules. Now let us have phenylketonuria. It is caused by a mutant recessive allele on chromosome 12. The affected individual lack an enzyme that is phenylalanine hydroxylase that catalyzes the conversion of the amino acid phenylalanine into tyrosine. Consequently, the phenylalanine amino acid is metabolized into phenylalanine pyruvate or phenylalanine pyruvic acid and other derivatives. Accumulation of these chemicals in the brain results in mental retardation. These are also excreted in urine as they are not absorbed by the kidney. Okay, now let us have chromosomal disorders. As we have learned, chromosomal disorder is of 2 types: structural disorder and numerical disorder. Again, numerical disorder is of 2 types: aneuploidy and polyploidy. Aneuploidy is again of 2 types: monosomy and trisomy. One example of trisomy is Down syndrome or Mongolian idiocy or Mongolism. It is caused by the presence of extra copy of the chromosome 21, that is trisomy of 21st chromosome. Due to this, the individual will have 47 chromosomes instead of 46 chromosomes. This disorder was first reported by Langdon Don in 1866. The defected individual will have partially open mouth with furrowed tongue, broad flat face with slanting eyes, broad palm with characteristic palm crease, small and round arched palate, epicanthal eye fold, congenital heart diseases, mental retardation. Okay, now let us have Klinefelter syndrome. Klinefelter syndrome is a chromosomal disorder which is caused by the presence of an extra X chromosome in the male, that is XXY. The individual has 47 chromosomes. It is also one example of trisomy. Though the individual is male, he shows a number of feminist characters. Some symptoms of an affected individual are tall stature with feminist physique, breast development which is called gynecomastia, female type of pubic hair pattern, poor beard growth, sterile. These are some characteristic features of Klinefelter syndrome. Now let us have Turner syndrome. Turner syndrome is caused due to the absence of one of the X chromosomes in a female. The karyotype or nucleus has only 45 chromosomes and they are called as X0 or XO females. The symptoms are web neck, rudimentary ovaries, poor development of breast, shell-shaped thorax, sterility. Okay, now let us learn about pedigree analysis. It is an analysis of traits in several generations of family chart. Pedigree analysis. It is an analysis of traits in several generations of family. Symbols used in pedigree analysis are. Let us know importance of pedigree analysis. Firstly, generalization period is long in human being, 20 to 25 years. Secondly, in many cases, human traits are influenced by a number of factors. Progeny of a couple is small. Thirdly, human beings have a mixed population and pure traits are rarely observed. In fourth, marriages desired by an experimenter are also always impossible in human beings. In pedigree analysis, we can study all types of Mendelian disorders, but chromosomal disorders are not studied in pedigree analysis. Why? Because chromosomal disorders are not inherited to their young ones. Okay, students, I hope you have understood the chapter. We have completed the chapter. If you have any doubt, you can ask questions. Thank you all. Have a nice day.